Section 18.3, Human Reproduction. By the end of this section, you will be able to describe human testicular and ovarian reproductive anatomies, describe spermatogenesis and oogenesis, and describe their differences and similarities, describe the role of hormones in human reproduction, and describe the roles of reproductive hormones. As in all animals, the adaptations for reproduction in humans are complex. They involve specialized and different anatomies in the two sexes, or a hormone regulation system, and specialized behaviors regulated by the brain and endocrine system. Human reproductive anatomy. The reproductive tissues of male and female humans develop similarly in utero until about the 7th week of gestation, when, in some cases, a low level of the hormone testosterone is released from the gonads. Testosterone causes the primitive gonads to differentiate into sexual organs, such as the scrotum and penis. When testosterone is absent, the primitive gonads develop into ovaries. Tissues that produce a penis in males produce a clitoris in females. The tissue that will become the scrotum in a male becomes the labia in a female. Thus, the male and female anatomies arise from a divergence in the development of what were once common embryonic structures. Male reproductive anatomy. Sperm are immobile at body temperature. Therefore, the testes are external to the body so that a correct temperature is maintained for motility. In land animals, including humans, the pair of testes must be suspended outside the body so the environment of the sperm is about 2 degrees Celsius lower than body temperature to produce viable sperm. If the testes do not descend through the abdominal cavity during fetal development, the individual has reduced fertility. The scrotum houses the testicles, or testes, singular testis, and provides passage for blood vessels, nerves, and muscles related to testicular function. The testes are a pair of male gonads that produce sperm and reproductive hormones. Each testis is approximately 2.5 by 3.8 centimeters, 1.5 by 1 inch, in size, and divided into wedge-shaped lobes by septa. Coiled in each wedge are seminiferous tubules that produce sperm. The penis drains urine from the urinary bladder and is a copulatory organ during intercourse. The penis contains 3 tubes of erectile tissue that become engorged with blood, making the penis erect in preparation for intercourse. The organ is inserted into the vagina, culminating with an ejaculation. During orgasm, the accessory organs and glands connected to the testes contract and empty the semen, containing sperm, into the urethra, and the fluid is expelled from the body by muscular contractions causing ejaculation. After intercourse, the blood drains from the erectile tissue and the penis becomes flaccid. Semen is a mixture of sperm, about 5% of the total, and fluids from accessory glands that contribute most of the semen's volume. Sperm are haploid cells consisting of a flagellum for motility, a neck that contains the cell's energy-producing mitochondria, and a head that contains the genetic material. An acrosome, acrosomal vesicle, is found at the top of the head of the sperm. This structure contains enzymes that can digest the protective coverings that surround the egg and allow the sperm to fuse with the egg. An ejaculate will contain from 2 to 5 milliliters of fluid and from 50 to 120 million sperm per milliliter. Figure 18.11. As seen in this scanning electron micrograph, human sperm has a flagellum, neck, and head. Sperm form in the walls of seminiferous tubules that are coiled inside the testes. The walls of the seminiferous tubules are made up of the developing sperm cells, with the least developed sperm at the periphery of the tubule and the fully developed sperm next to the lumen. The sperm cells are associated with Sertoli cells that nourish and promote the development of the sperm. Other cells present between the walls of the tubules are the interstitial cells of Leydig, which produce testosterone once the male reaches adolescence. When the sperm have developed flagella, they leave the seminiferous tubules and enter the epididymis. This structure lies along the top and posterior of the testes and is the site of sperm maturation. The sperm leave the epididymis and enter the vas deferens, which carries the sperm behind the bladder and forms the ejaculatory duct with the duct from the seminal vesicles. During a vasectomy, a section of the vas deferens is removed, preventing sperm, but not the secretions of the accessory glands, from being passed out of the body during ejaculation and preventing fertilization. The bulk of the semen comes from the accessory glands associated with the male reproductive system. These are the seminal vesicles, the prostate gland, and the bulbourethral gland. The secretions from the accessory glands provide important compounds for the sperm, including nutrients, electrolytes, and pH buffering. There are also coagulation factors that affect sperm delivery and motility. Figure 18.12. The testicular reproductive system structures are shown. Table 18.1. Testicular reproductive system anatomy. Female reproductive anatomy. A number of female reproductive structures are exterior to the body. These include the breast and the vulva, which consist of the mons pubis, clitoris, labia majora, labia minora, and the vestibular glands. Figure 18.13. The structures of the human ovarian reproductive system are shown. The breasts consist of mammary glands and fat. Each gland consists of 15 to 25 lobes that have ducts that empty at the nipple and that supply the nursing child with nutrient and antibody-rich milk to aid development and protect the child. Internal female reproductive structures include ovaries, oviducts, the uterus, and the vagina. The pair of ovaries is held in place in the abdominal cavity by a system of ligaments. The outermost layer of the ovary is made up of follicles, each consisting of one or more follicular cells that surround, nourish, and protect a single egg. During the menstrual period, a batch of follicular cells develops and prepares their eggs for release. At ovulation, one follicle ruptures and one egg is released. Following ovulation, the follicular tissue that surrounds the ovulated egg stays within the ovary and grows to form a solid mass called the corpus luteum. The corpus luteum secretes additional estrogen and the hormone progesterone that helps maintain the uterine lining during pregnancy. The ovaries also produce hormones such as estrogen. The oviducts, or fallopian tubes, extend from the uterus in the lower abdominal cavity to the ovaries, but they are not in contact with the ovaries. The lateral ends of the oviducts flare out into a trumpet-like structure and have a fringe of finger-like projections called fimbriae. When an egg is released at ovulation, the fimbriae help the non-motile egg enter into the tube. The walls of the oviducts have a ciliated epithelium over smooth muscle. The cilia beat and the smooth muscle contracts, moving the egg toward the uterus. Fertilization usually takes place within the oviduct, and the developing embryo is moved toward the uterus. It usually takes the egg or embryo a week to travel through the oviduct. Sterilization in females is called a tubal ligation. It is analogous to a vasectomy in males in that the oviducts are severed and sealed, preventing sperm from reaching the egg. The uterus is a structure about the size of a person's fist. The uterus has a thick muscular wall and is lined with an endometrium rich in blood vessels and mucus glands that develop and thicken during the female cycle. Thickening of the endometrium prepares the uterus to receive the fertilized egg or zygote, which will then implant itself in the endometrium. The uterus supports the developing embryo and fetus during gestation. Contractions of the smooth muscle in the uterus aid in forcing the baby through the vagina during labor. If fertilization does not occur, a portion of the lining of the uterus sloughs off during each menstrual period. The endometrium builds up again in preparation for implantation. Part of the uterus, called the cervix, protrudes into the top of the vagina. The vagina is a muscular tube that serves several purposes. It allows menstrual flow to leave the body. It is the receptacle for the penis during intercourse and the pathway for the delivery of offspring. Table 18.2, Ovarian Reproductive System Anatomy. Gametogenesis, spermatogenesis, and oogenesis. Gametogenesis, the production of sperm and eggs, involves the process of meiosis. During meiosis, two nuclear divisions separate the paired chromosomes in the nucleus and then separate the chromatids that were made during an earlier stage of the cell's life cycle. Meiosis and its associated cell divisions produces haploid cells with half of each pair of chromosomes normally found in diploid cells. The production of sperm is called spermatogenesis, and the production of eggs is called oogenesis. Spermatogenesis. Spermatogenesis occurs in the wall of the seminiferous tubules, with the most primitive cells at the periphery of the tube and the most mature sperm at the lumen of the tube. Immediately under the capsule of the tubule are diploid, undifferentiated cells. These stem cells, each called a spermatogonium, plural spermatogonia, go through mitosis to produce one cell that remains as a stem cell and a second cell called a primary spermatocyte that will undergo meiosis to produce sperm. The diploid primary spermatocyte goes through meiosis 1 to produce two haploid cells called secondary spermatocytes. Each secondary spermatocyte divides after meiosis 2 to produce two cells called spermatids. The spermatids eventually reach the lumen of the tubule and grow a flagellum, becoming sperm cells. 4 sperm result from each primary spermatocyte that goes through meiosis. Figure 18.14. During spermatogenesis, 4 sperm result from each primary spermatocyte. The process also maps onto the physical structure of the wall of the seminiferous tubule, with the spermatogonia on the outer side of the tubule, and the sperm with their developing tails extended into the lumen of the tubule. Oogenesis. Oogenesis occurs in the outermost layers of the ovaries. As with sperm production, oogenesis starts with the germ cell. In oogenesis, This this germ cell is called an oogonium and forms during the embryological development of the individual. The oogonium undergoes mitosis to produce about 1 to 2 million oocytes by the time of birth. Figure 18.15. The process of oogenesis occurs in the ovary's outermost layer. The primary oocytes begin meiosis before birth. However, the meiotic division is arrested in its progress in the first prophase stage. At the time of birth, all future eggs are in prophase 1. This situation is in contrast with the testicular reproductive system, in which sperm are produced continuously throughout the life of the individual. Starting at adolescence, anterior pituitary hormones cause the development of a few follicles in an ovary each month. This results in a primary oocyte finishing the first meiotic division. The cell divides unequally, with most of the cytoplasm and organelles going to one cell, called a secondary oocyte, and only one set of chromosomes, and a small set of cytoplasm going to the other cell. This second cell is called a polar body and usually dies. Cell division is again arrested, this time at metaphase 2. At ovulation, this secondary oocyte is released and travels toward the uterus through the oviduct. If the secondary oocyte is fertilized, the cell continues through meiosis 2, producing a second polar body and haploid egg, which fuses with the haploid sperm to form a fertilized egg, a zygote, containing all 46 chromosomes. Hormonal control of reproduction. The human reproductive cycles are controlled by the interaction of hormones from the hypothalamus and anterior pituitary with hormones from reproductive tissues and organs. The hypothalamus monitors and causes the release of hormones from the anterior pituitary gland. When the reproductive hormone is required, the hypothalamus sends a gonadotropin-releasing hormone, GnRH, to the anterior pituitary. This causes the release of follicle-stimulating hormone and luteinizing hormone, FSH and LH, from the anterior pituitary into the blood. Although these hormones are named after their functions in female reproduction, they are produced in both sexes and play important roles in controlling reproduction. Other hormones have specific functions in the male and female reproductive systems. Male hormones. At the onset of puberty, the hypothalamus causes the release of FSH and LH into the male system for the first time. FSH enters the testes and stimulates the Sertoli cells located in the walls of the seminiferous tubules to begin promoting spermatogenesis. LH also enters the testes and stimulates the interstitial cells of Leydig, located in between the walls of the seminiferous tubules, to make and release testosterone into the testes and the blood. Testosterone stimulates spermatogenesis. During adolescence, this hormone is also responsible for a deepening of the voice, the growth of facial, axillary, and pubic hair, an increase in muscle bulk, and the beginnings of the sex drive. Figure 18.16. Hormones control sperm production in a negative feedback system. A negative feedback system occurs in the male with rising levels of testosterone acting on the hypothalamus and anterior pituitary to inhibit the release of GnRH, FSH, and LH. In addition, the Sertoli cells produce the hormone inhibin, which is released into the blood when the sperm count is too high. This inhibits the release of GnRH and FSH, which will cause spermatogenesis to slow down. If the sperm count reaches a low of 20 million per milliliter, the Sertoli cells cease the release of inhibin and the sperm count increases. Female hormones. The control of reproduction in females is more complex. The female reproductive cycle is divided into the ovarian cycle and the menstrual cycle. The ovarian cycle governs the preparation of endocrine tissues and release of eggs, while the menstrual cycle governs the preparation and maintenance of the uterine lining. These cycles are coordinated over a 22 to 32 day cycle, with an average length of 28 days. As with the male, the GnRH from the hypothalamus causes the release of the hormones FSH and LH from the anterior pituitary. In addition, estrogen and progesterone are released from the developing follicles. As with testosterone in males, estrogen is responsible for the secondary sexual characteristics of females. These include breast development, flaring of the hips, and a shorter period for bone growth. The ovarian cycle and the menstrual cycle. The ovarian and menstrual cycles are regulated by hormones of the hypothalamus, pituitary, and ovaries. The ebb and flow of the hormones causes the ovarian and menstrual cycles to advance. The ovarian and menstrual cycles occur concurrently. The first half of the ovarian cycle is the follicular phase. Slowly rising levels of FSH cause the growth of follicles on the surface of the ovary. This process prepares the egg for ovulation. As the follicles grow, they begin releasing estrogen. The first few days of this cycle coincide with menstruation, or the sloughing off of the functional layer of the endometrium in the uterus. After about 5 days, estrogen levels rise and the menstrual cycle enters the proliferative phase. The endometrium begins to regrow, replacing the blood vessels and glands that deteriorated during the end of the last cycle. Figure 18.17. The ovarian and menstrual cycles of reproduction are regulated by hormones produced by the hypothalamus, pituitary, and ovaries. Just prior to the middle of the cycle, approximately day 14, the high level of estrogen causes FSH and especially LH to rise rapidly and then fall. The spike in LH causes the most mature follicle to rupture and release its egg. This is ovulation. The follicles that did not rupture degenerate and their eggs are lost. The level of estrogen decreases when the extra follicles degenerate. Following ovulation, the ovarian cycle enters its luteal phase and the menstrual cycle enters its secretory phase, both of which run from about day 15 to 28. The luteal and secretory phases refer to changes in the ruptured follicle. The cells in the follicle undergo physical changes and produce a structure called a corpus luteum. The corpus luteum produces estrogen and progesterone. The progesterone facilitates the regrowth of the uterine lining and inhibits the release of further FSH and LH. The uterus is being prepared to accept a fertilized egg should it occur during the cycle. The inhibition of FSH and LH prevents any further eggs and follicles from developing, while the progesterone is elevated. The level of estrogen produced by the corpus luteum increases to a steady level for the next few days. If no fertilized egg is implanted into the uterus, the corpus luteum degenerates and the levels of estrogen and progesterone decrease. the endometrium begins to degenerate as the progesterone levels drop, initiating the next menstrual cycle. The decrease in progesterone also allows the hypothalamus to send GnRH to the anterior pituitary, releasing FSH and LH and starting the cycles again. Career connection: reproductive endocrinologist. A reproductive endocrinologist is a physician who treats a variety of hormonal disorders related to reproduction and infertility in people of any gender. The disorders include menstrual problems, infertility, pregnancy loss, sexual dysfunction, and menopause. Doctors may use fertility drugs, surgery, or assisted reproductive techniques, ART, in their therapy. ART involves the use of procedures to manipulate the egg or sperm to facilitate reproduction, such as in vitro fertilization. Reproductive endocrinologists undergo extensive medical training, first in a 4-year residency in obstetrics and gynecology, then in a 3-year fellowship in reproductive endocrinology. To be board certified in this area, the physician must pass written and oral exams in both areas. Gestation. Pregnancy begins with the fertilization of an egg and continues through to the birth of the individual. The length of time of gestation, or the gestation period, in humans is 266 days and is similar in other great apes. Within 24 hours of fertilization, the egg nucleus has finished meiosis and the egg and sperm nuclei fuse. With fusion, the cell is known as a zygote. The zygote initiates cleavage and the developing embryo travels through the oviduct to the uterus. The developing embryo must implant into the wall of the uterus within 7 days or it will deteriorate and die. The outer layers of the developing embryo, or blastocyst, grow into the endometrium by digesting the endometrial cells, and healing of the endometrium closes up the blastocyst into the tissue. Another layer of the blastocyst, the chorion, begins releasing a hormone called human beta chorionic gonadotropin, or beta HCG, which makes its way to the corpus luteum and keeps the structure alive, or active. This ensures adequate levels of progesterone that will maintain the endometrium of the uterus for the support of the developing embryo. Pregnancy tests determine the levels of beta HCG in urine or serum. If the hormone is present, the test is positive. The gestation period is divided into 3 equal periods or trimesters. During the first 2 to 4 weeks of the first trimester, nutrition and waste are handled by the endometrial lining through diffusion. As the trimester progresses, the outer layer of the embryo begins to merge with the endometrium, and the placenta forms. The placenta takes over the nutrient and waste requirements of the embryo and fetus, with the gestational parent's blood passing nutrients to the placenta and removing waste from it. Chemicals from the fetus, such as bilirubin, are processed by the gestational parent's liver for elimination. Some of the pregnant person's immunoglobulins will pass through the placenta, providing passive immunity against some potential infections. Internal organs and body structures begin to develop during the first trimester. By 5 weeks, limb buds, eyes, the heart, and liver have been basically formed. By 8 weeks, the term fetus applies and the body is essentially formed. The individual is about 5 centimeters or 2 inches in length, and many of the organs, such as the lungs and liver, are not yet functioning. Exposure to any toxins is especially dangerous during the first trimester, as all of the body's organs and structures are going through initial development. Anything that interferes with chemical signaling during that development can have a severe effect on the fetus's survival. Figure 18.18. A. Fetal development is shown at 9 weeks gestation. B. This fetus is just entering the second trimester, when the placenta takes over more of the functions performed as the baby develops. C. There is rapid fetal growth during the third trimester. During the second trimester, the fetus grows to about 30 centimeters, or about 12 inches. It becomes active, and the pregnant person usually feels the first movements. All organs and structures continue to develop. The placenta has taken over the functions of nutrition and waste elimination and the production of estrogen and progesterone from the corpus luteum, which has degenerated. The placenta will continue functioning up through the delivery of the baby. During the third trimester, the fetus grows to 3 to 4 kilograms, 6.5 to 8.5 pounds, and about 50 centimeters, or 19 to 20 inches long. This is the period of the most rapid growth during the pregnancy, as all organ systems continue to grow and develop. Labor is the muscular contractions to expel the fetus and placenta from the uterus. Toward the end of the third trimester, estrogen causes receptors on the uterine wall to develop and bind the hormone oxytocin. At this time, the baby reorients, facing forward and down with the back or crown of the head engaging the cervix, the uterine opening. This causes the cervix to stretch, and nerve impulses are sent to the hypothalamus, which signals the release of oxytocin from the posterior pituitary. Oxytocin causes smooth muscle in the uterine wall to contract. At the same time, the placenta releases prostaglandins into the uterus, increasing the contractions. A positive feedback relay occurs between the uterus, hypothalamus, and the posterior pituitary to assure an adequate supply of oxytocin. As more smooth muscle cells are recruited, the contractions increase in intensity and force. There are 3 stages to labor. During stage 1, the cervix thins and dilates. This is necessary for the baby and placenta to be expelled during birth. The cervix will eventually dilate to about 10 centimeters. During stage 2, the baby is expelled from the uterus. The uterus contracts, and in most cases, the person giving birth pushes as they compress the abdominal muscles to aid the delivery. The last stage is the passage of the placenta after the baby has been born and the organ has completely disengaged from the uterine wall. If labor should stop before stage 2 is reached, synthetic oxytocin, known as pitocin, can be administered to restart and maintain labor.